Our next panelist slash presenter, Dr. Daria Popugaeva, was a consultant, is a consultant, I should say. So is a research engineer at Western University and a water quality specialist at Geoscience Services. Daria holds a PhD in environmental engineering from Western University, and I'm very excited about this talk because for two reasons. First of all, the consulting world gets into research, and I think there is a lot to learn from that collaboration. And secondly, in my world with the bacteria and in Taylor's world, we're spending our time trying to defeat the cold when you'll see they are using the cold to their advantage. So please help me in welcoming Daria.
Thank you very much, Guillaume, for the introduction, and hello, everyone. So the topic of my presentation is dedicated to a project when we focused on the water treatment approach that combines two green methods, and those are cryopurification, this is basically freezing of the water, and a microbial fuel cell process. Our focus is on the development and optimization of this process. Especially, we are focusing more on the freezing technology, and I would like to highlight why we do that. First of all, green technology. We don't use any chemicals. This is a process that relies on physics only. And of course, when we talk about the cold climates, this is when the beauty of this process shines, because cold climates is basically cold temperatures, and this is a driving force for our process to start treating the water. So our focus is on the ferromine water that is highly contaminated with zinc concentrations. And our goal is to decrease these concentrations of zinc to the quality standard for that, and it's 0.5 milligram per liter. So to do that, we use the following two methods, and that includes laboratory study and a mathematical modeling. So what we do, we have a laboratory setup for both of the methods. We have a circulating chiller for the cryopurification. We designed the vessels that we used, and we filled out those vessels with the ferromine water. And for the microbial fuel cell process, we use the following bacteria. And what we did as well is we simulate this process of cryopurification. And to do that, a commercially available software was used, and the following three models were used to simulation purposes. So let's take a look to the diagram of the setup and how these two methods combines into one approach. So we have two phases. Phase one, we have our ferromine water that we treat through cryopurification, and we produce pure ice and the more concentrated solution, so-called brine. And this brine goes to the second stage of the process, where we have the microbial fuel cell process. So I will go straight to the results and start with the cryopurification results. Freezing of the water. Once again, no chemicals. This is only physics. And I would like to highlight the advantages of that when we talk about cold climates, cold temperatures. So when we started working on the laboratory experiments, the first question came in mind: what if we change the ice shapes? Will it affect the result? Will it affect the efficiency of zinc removal? And to do that, we have our Mine water. This is MIW in those slides, and we freeze it from different directions to produce different ice shapes. And then change the temperature. So this is the result when we have zinc removal as a function of freezing temperature and the function of different ice shapes. And the answer to our question, if the ice shape has a difference, yes, it is. When we freeze it from the top, we will have the highest zinc removal. And the higher the temperature, the higher the zinc removal. So with these experiments, we were able to achieve up to 80% removal when we freeze it from the top at the temperature of -1. Of course, cold climates, we need to go to the lower temperature, and we wanted to find a way how to improve the removal. This is when we introduced mixing to the experiments, and that is a mechanical mixing, extra energy, of course, but still what it gave us a better removal we were able to achieve up to 65% at minus 15 degrees and up to 95% of zinc removal using all of the vessels that we use. And this 95% removal of zinc gives us a chance to meet our objective to decrease zinc concentration to the close to the effluent quality standard. So why we did that? Why we did all of these experiments? The reason behind this, because we are now completing the laboratory test study, and our next step is scaling up the reactor. We needed to know what are the key parameters that we need to consider when we upscaling the reactor. And we highlighted a few of them, and one of the main one is freezing temperature, and of course mixing. And with that, using only one cycle of freezing, we can achieve up to 95% of zinc removal. So, mixing, we used mechanical mixing and extra energy. We wanted to remove that factor. We wanted to decrease the energy usage and of course the cost for the experiments. And that is when we started to think what we can do. An idea that came in mind is a natural convection, and those slides, the next few slides, is the result of the natural convection. So unfortunately, one of the video it doesn't work, but what it shows here. So the natural convection, this is physical phenomenon that defines a lot of on our planet, starting from the weather temperature and the conditions of the ocean. So you can. So this one doesn't work, but what is here? So we freeze it from the top during the time. So this is a simulation results, and the water starts to mix, and this is the velocity profile for exact same results. So water inside of our vessel mixes naturally. Also, the results of mathematical model that we developed showed us how we can combine these two. So here is the temperature and the velocity. So the vectors is the velocity and the colors is the temperature change. And you can see how it mixes during the, in this case, 5 minutes of real time. And we validated our model comparing by our results of experiments and the results of simulation. Also, when we talk about freezing and we talk about ice formation, we have to understand what's going on with the impurity. And on this slide, this is the simulation of ice formation when we freeze it from the walls. And the more concentrated water goes down to the bottom, and you can see this darker color. This is basically our contaminant that goes down. And the dark blue, this is pure ice. So, the purer the ice we have, the more concentrated the brine solution. And of course, we cannot just dispose the brine. We don't want to harm the environment. So we need to treat the brine. And our approach to treat the brine is to use another green technology that is a microbial fuel cell process. It also has a number of advantages, and one of them, it can, under optimal conditions, it can generate electricity. And just a quick overview of the results on the Microbial fuel cell process. We ran a few experiments, and 2 of them were using the brine solution, and 1 of them using the feed water from the ferromine. And the results of experiments show that under 180 hours, 20 hours, our bacteria can absorb approximately 93% of zinc. But the highest activity occurs within the first 24 hours. So with that, we can also achieve the concentrations of zinc that we're targeting. This is wrap up the part for the ferromine water. Then we made an assumption in our experiments and in our process of freezing the water, of cryopurification. So can we apply this technology to different water types, to different water bodies, different contaminants? And to address this question, we ran a set of experiments using the water coming from the Wolverine mine, and the parameter that we chose was selenium. And we knew the optimal conditions for this cryopurification experiments, and we noticed that, yes, the technology is efficient and can be applied to different water types. So yes, I would like to wrap up my presentation with the main conclusions and future steps within the project. So we tested this technology, this approach, and we found this effective. We highlighted the main parameters that are the most important during the purification process, and this is the foundation for us to move to the next step, to the bench scale testing and to the pilot scale testing. And our goal is to incorporate the machine learning techniques into the pilot scale testing so that we can optimize and make the process even more efficient. Our research received an interest from different sources, and our future steps is to, of course, to build new partnership, to work on this project and to advance this project. The results of our work were published, and for those who are interested, please let me know. I can share this paper with you. And yes, and I just want to acknowledge my colleagues from Core Geoscience Services and from Western University, and thank our financial sponsors from INSERC and MyTax. So with that, thank you so much for your interest, and I'm open for questions.
Well, there's all sorts of them, but I'll try that one.
When we've been talking actually right before, we talked about the challenges of trying to freeze and eventually tried using spraying and also failed and pipe scale. How are you planning on addressing that at pilot scale?
So pilot scale. So what we need to do before the pilot scale, this is a lab test and bench scale test, and this is when we also will check our equipment in those conditions, including for the lower temperatures. This is what we discussed, right? The equipment failure when it comes to really low temperature outside, especially in Yukon. So yeah, we plan to address these questions in the bench scale before we move to the pilot scale.
I guess a similar question
Sure.
on the freezing part of your work.
Mm-hmm.
So how do you deal with supercooling? Because clearly you're in that density phase or the temperature phase for water. You know, because clearly when you're at supercooling, you have to deal with frazzle ice and with slush ice as a minimum, those two types of ice. And I think there's some other ones too, but those are the two I'm familiar with.
Thank you for your question. That's a really good one. So we dealt with the supercooling in our experiments, especially if we are talking about temperatures of -1 and up, like we did it at -1 and -0.5 degrees. And we noticed that slushy ice, fraser ice formation, fraser ice first and then slushy. So what we did for that, we just filtered that part. And yes, this is what was the approach. But what we also notice, when we have the fraser ice, we collect that ice, and we have slushy ice, we also collect that. And we measured the concentration, how pure is that? And the results show that this is the purest ice. Yes. So our thought was, should we focus more on the fraser ice, for example, instead of the bulk ice? Unfortunately, I would say no, because it takes a lot of time to produce a certain amount of this fraser ice in order to, to have enough volume to even measure the concentration. So that's not an option. And if we're talking about lower temperature, like -5 or -10, there will be no supercooling. And also, in terms of supercooling, because temperature goes down below 0 and there is no ice. This is a supercooling thing. And what we did to address that, we added seed ice to speed up the process. So once you added the seed ice to the supercooled water, ice will start form instantly. And this is your bulk ice, so basically what we wanted.
No more question? I have one last. When I was a consultant, I was frustrated about the lack of research in my consulting world.
Mm-hmm.
I saw the benefit of the collaboration. My question is, how do you work with Corgeo, with a consulting firm, and how do you make it a win-win situation?
Interesting question. So my academia experience is quite long. So I started with bachelor and finished master's, then PhD, and then postdoctoral fellowship. That was a lot of years, and I really wanted to enter consulting world for a number of reasons: to get new experience and to apply my knowledge and skills into the industrial projects. So how do we make it win-win situation? So we work on this research and development project together with Corgeo, and it's amazing because we have various experts within the team, various expertise, various background, and this is beneficial in terms of the overall result. Yeah.
Thank you.
Thank you.
So we talked about remediation, which is an interesting world about how can we treat contamination, remove a contamination from somewhere. Restoration is at least as important, which is how can you bring, for example, a disturbed site to look good or to be as it was before the disturbance. The next panelist / presenter is not in the Yukon right now. She couldn't make it, but Dr. Catherine Stewart sent us her presentation. It's a small video, it's 10 minutes long. Catherine Stewart, Dr. Stewart, is an associate professor at Soil Science Department at U of S. Her research areas are restoration, ecology, remediation, northern plant soil systems. She worked a lot in the Yukon. I actually met her decades ago at Yukon College at the time, and she's going to give us a very good introduction to the talks that will follow about mine revegetation. So please welcome virtually Catherine Stewart, I guess.